Hello, everyone, and welcome to our podcast on artificial intelligence and drug discovery in infectious diseases. This podcast explores how cutting edge computational tools are reshaping the way we understand, develop, and deploy therapeutics, particularly in the context of infectious diseases. So, let's get started. The identification of drug targets is the first and most critical step in the process of drug discovery. For decades, this phase relied on hypothesis driven experimentation that could take years to yield actionable insights. Today, artificial intelligence is revolutionizing this space by enabling the systemic analysis of vast molecular data sets to uncover therapeutic vulnerabilities in pathogens. AI algorithms can process large volumes of genomic, proteomic, and transcriptomic data, allowing researchers to pinpoint specific genes or proteins that are essential for a pathogen's survival, replication, or virulence. Machine learning models can then rank these targets based on druggability, safety, and evolutionary conservation, helping to prioritize candidates with the greatest therapeutic potential. What is particularly transformative is AI's ability to recognize complex patterns in biological networks, identifying functional relationships that may not be evident through traditional methods. This includes mapping host pathogen interactions, identifying co regulated gene clusters, and detecting molecular signatures specific to resistant strains. These insights enhance precision and reduce the trial and error aspects of early stage research. Furthermore, AI enables in silico validation of targets through simulations and predictive modeling. This allows researchers to anticipate how a drug might interact with a target or whether off target effects are likely to occur, thereby reducing the dependency on early animal models and high throughput screens. The COVID 19 pandemic offered a real time validation of this approach. Several AI platforms were used to scan SARS-CoV-2 proteins and prioritize structural components like the spike protein for vaccine and antiviral design. These predictions, generated within days of the genome being released, dramatically shortened the lead time for experimental work. Looking forward, the integration of AI with CRISPR screening, single cell sequencing, and patient derived data is expected to further improve target discovery, especially for complex or emerging infectious diseases. As datasets become richer and more global in scope, AI driven target identification is poised to become an indispensable pillar of infectious disease research.
I'll talk a little bit about repurposing existing drugs for infectious diseases using AI now. Now, drug repurposing using existing drugs for new therapeutic indications, it's not a novel concept, but artificial intelligence has given it a new momentum. By reducing both the cost and time required to bring therapies to market, AI enabled repurposing offers a practical solution to urgent health crises, particularly in the realm of infectious diseases. At the heart of the AI driven repurposing is the ability to detect non obvious relationships between drugs and disease mechanisms. Using data from clinical trials, electronic health records, and molecular databases, AI can identify patterns and correlations that suggest potential off label uses for approved medications. Even during the COVID 19 pandemic, AI platforms were rapidly deployed to screen thousands of FDA approved drugs against SARS CoV 2 proteins. Several drugs, including remdesivir and baricitinib, were flagged early on as potential candidates. While not all such predictions translated into clinical success, the speed and efficiency with which they were identified underscored the power of algorithmic screening. Importantly, repurposing doesn't only involve antivirals. AI models have suggested roles for immunomodulators, anticoagulants, and even anti inflammatory agents in treating infectious diseases, particularly those with a strong host response component. This multifactorial perspective is crucial for diseases like sepsis, dengue, or COVID 19, where the pathology is driven as much by the immune system as by the pathogen. The advantages are particularly relevant for developing nations. In countries like India, where access to novel therapeutics can be delayed by years due to regulatory, economic, or logistical barriers, the ability to repurpose affordable, widely available drugs is invaluable. Generic manufacturing infrastructure further enhances this feasibility, allowing for rapid deployment if supported by robust evidence. However, the success of AI driven repurposing depends on the availability of high quality integrated datasets. As more open source initiatives emerge to share pharmacological, clinical, and molecular data, AI's predictive accuracy will continue to improve, enabling evidence based repurposing at scale. Ultimately, AI is making drug repurposing not only faster but smarter by guiding researchers toward the most promising candidates based on data driven insights. It ensures that efforts are directed where they're most likely to yield meaningful therapeutic impact.
Today, I'm going to discuss about generative AI for designing novel therapeutics and peptides. Generative AI is redefining how we approach drug discovery. Using transformer models and generative adversarial networks, researchers can now design entirely new classes of molecules, antimicrobial peptides, enzyme inhibitors, or even synthetic proteins tailored to specific pathogens. These models learn the structure activity patterns of known compounds and generate novel candidates with improved efficacy and safety profiles. Transformer models, once exclusive to natural language processing, have been adapted to molecular design by treating chemical structures as sequences. This enables the generation of synaptically valid molecules with desirable biological properties. GANs take it a step further, pitting a molecule generator against a discriminator, refining outputs through competition until they closely resemble effective therapeutics. The value of these methods lies in the speed and diversity of output. Thousands of structurally novel molecules can be generated, filtered, and prioritized for synthesis in a fraction of the time needed for traditional medicine chemistry. Many of these AI suggested molecules are proving effective against resistant bacteria or as antiviral agents. These tools are also highly customizable. They can be trained on specific pathogen features, such as the viral protease in dengue or the outer membrane of gram negative bacteria, to design inhibitors that selectively bind to those targets. This is especially important in the context of rising antimicrobial resistance. With increasing integration into existing drug discovery workflows, generative AI is not just supplementing human creativity, it is offering entirely new chemical space to explore. As validation pipelines improve, these tools could form the backbone of future antimicrobial development. Thank you.
Vaccine development has significantly accelerated with AI. Deep learning models are being used to identify immunogenic epitopes, optimize mRNA sequences, and even predict future viral mutations, all crucial in designing effective and timely vaccines for rapidly evolving pathogens like SARS-CoV-2 or influenza. Epitope prediction models analyze viral proteins and human immune responses to identify regions most likely to trigger neutralizing antibodies. This ensures that the vaccine targets components of the virus with the greatest immunological relevance, reducing the risk of escape variants. AI also enhances mRNA vaccine design by improving codon usage, stability, and translational efficiency. These optimizations contribute to strong and more sustained immune responses, which are particularly important when rolling out vaccines under emergency use authorizations. Another crucial role of AI is forecasting viral evolution. By analyzing mutation patterns and host interactions, machine learning models can suggest which variants are likely to become dominant. This insight helps researchers preemptively adjust vaccine compositions, a critical strategy for influenza and potentially universal coronavirus vaccine. AI further aids in identifying conserved regions across viral families, supporting the design of pan pathogen vaccines. These approaches may reduce the need for frequent booster updates and are particularly useful for low resource setting where logistic challenge continues vaccination efforts. In sum, AI is enabling a shift from reactive to predictive vaccine design, one that anticipates viral behavior and enhances global preparedness.
Accurately forecasting disease outbreaks is key to proactive drug development and public health readiness. AI offers powerful tool to analyze real-time surveillance data, environmental variables, and human mobility to predict where and when the next infectious disease hotspot might emerge. Machine learning models integrate satellite data, weather pattern, land use, and socio-economic factors to identify conditions favorable for disease spread, such as vector-borne diseases linked to rainfall or zoonotic spillovers associated with deforestation. These models provide early warnings for conditions that could lead to epidemics. AI also tracks and models human behavior using travel data, population density, and social media signals to predict how infection might spread across regions or countries. These insights can inform containment strategies and drug deployment planning. Notable platforms like Blue Dot Health Map have demonstrated AI's potential by identifying outbreaks days before official recognition. Such tools are becoming increasingly vital as urbanization and climate change alter the dynamics of disease emergence. In drug discovery context, knowing the likely geography and timing of an outbreak helps researchers align compound screening, clinical trial sites, and manufacturing priorities. For diseases with seasonality or regional specificity, this alignment is crucial. For developing countries, especially, AI driven forecasting supports more targeted use of limited public health resources and enable early intervention, reducing both disease burden and healthcare costs.
Clinical trial acceleration using AI. Clinical trials are often the most time consuming and expensive part of drug development. In the context of infectious diseases, where rapid response is critical, artificial intelligence is increasingly being used to accelerate each stage of the trial process without compromising safety or rigor. AI supports smarter protocol design by analyzing past trial outcomes, patient demographics, and disease progression models. This helps optimize dosage schedules, inclusion criteria, and endpoints, thereby improving the likelihood of success. For emerging infectious diseases, this speed can be life saving. Patient stratification is another key area where AI makes a difference. By clustering patient data, such as genetics, comorbidities, and biomarker profiles, AI can help select the right subgroups most likely to respond to treatment. This is particularly vital in diverse or underrepresented populations. AI tools also assist in site selection by identifying geographic locations with suitable patient pools, clinical infrastructure, and logistical feasibility. This is essential for infectious disease trials, which often require rapid deployment in outbreak zones or resource limited settings. Once a trial is underway, AI supports real time monitoring of data and adverse events, flagging anomalies faster than traditional systems. This leads to better safety management, quicker adjustments, and more adaptive trial designs. Together, these AI driven efficiencies could substantially reduce the cost and time of bringing new antimicrobial agents and vaccines to market, while also improving inclusivity and scientific rigor.